Hi there, my name is John James. I'm an Associate Professor in the Warwick Medical School, and I am the module lead for Block 13 in the Integrated Science course. And my block is titled, How Can Non-Self Be Recognised? But in some ways, it's probably easier to think of this as how does our immune system function? What is it trying to do? How does it protect us and keep us healthy? And the video you should hopefully be seeing in front of you right now is a video of the lymph node, or the place where immune cells meet potentially infected cells inside a mouse. What I hope you can see is some bright green cells, and these bright green cells are the ones that are potentially infected with a virus or a bacterium or something like that. And the red cells running around are the T cells. And T cells are what I work on, and they're really one of our most important white blood cells, which I'll talk about more. So the job of these red T cells you can see in front of you is to try and find those green presenting cells, the ones that have been infected by some kind of virus. And you'll probably think that's really easy, it's the green cells, but obviously, they've been colored green for your benefit, but in reality, the T cells have to find those cells through some various means of using biochemistry or biophysics to both recognize them and respond to them. But beyond that, if you also tell, there's a lot of darkness in the image, and that's because there are lots and lots, literally thousands of cells that aren't infected. So not only do the T cells have to find the infected cells, they have to make sure they don't find the healthy ones, and that's really the key to this module. Is how does a T cell or any immune cell recognize non-self, foreign, compared to all the cells that are your natural cells. So what you get hopefully in this 2 weeks will be trying to understand, why do we actually need an immune system? I mean, can we get away without having an immune system? Are we unique in having an immune system? I'm sure you appreciate that we now realize through CRISPR-Cas9 that bacteria are also very, very good at having an adaptive immune system, which is even 10 years ago or so was not really a well-known fact. We'll describe the different parts of our immune system, both from what happens when you first get an infection, all the way through to its resolution and the memory of that response, hence the kind of vaccination or adaptive immune response, which, given the light of COVID-19 at the moment, is something that obviously is playing a lot of people's minds, is whether we can really have an acquired immunity against something like a coronavirus. We'll talk about how we make a response to potentially any infection. That's really our adaptive immune system, which I'll spend more time on. But not only that, is how clearly we do get poorly sometimes. Sometimes it's very deleterious, and the question is, how do bacteria and viruses, which are well known to infect us, how can they fight back? How do they trick our own immune systems so that they can win the race of keeping healthy? And also, sometimes our immune system goes wrong. Autoimmunity really is a classic example where the balance between recognizing healthy versus infected cells goes wrong. So that goes back to the whole title of the module, where what if non-self is treated as self? And that's really what autoimmunity is. So we'll also discuss what that means. So the immune system, you can study it in terms of purely as a sort of immunology-based science, but in reality, it actually really brings together a huge diverse array of different disciplines, all trying to understand how immune cells recognize what is you and what is not you, what has infected you. And so the things I work on tend to be more the sort of, as you can hopefully see on the screen, more the biochemistry and the biophysics of how immune cells recognize non-self. But there's a huge wealth of literature on using math and statistics as well to try and answer how our immune system works. These cells are, of course, also normal cells. There's nothing particularly special about them except they do this job in the immune system. So there's plenty of very interesting cell biology that also plays into this as well. So in some ways, I feel that the immune system, as hopefully I'll teach it for the 2 weeks, is a really good exemplar for why you need to think in an integrated way. That you need to bring in all these different facets of how cells are likely to function to really understand it, not only for the purely sense of great, we understand it, but also, as I'm sure you can appreciate, that a good understanding of how our immune system functions means we can work out ways to improve it when it does go wrong, and it does go wrong a fair amount of time for people. So probably the best way to go through what we'll discuss really is trying to understand what the labs will be about. And the first sort of part of the labs will just simply be to look at your immune system. Unfortunately, in good old ye olden days, you used to be able to just go around, grab a undergraduate student such as yourselves, and stab them and take some blood out. These days are sadly gone, but we can still get some human whole blood, and which I hope you can see. Maybe this even works. If you can see, let's ring that in blood red. This is whole human blood here, and it's red. I'm sure you all hopefully seen your blood at this point in your lives, but you can, inside that blood is really everything that your immune system does and has to fight an infection, which I think is amazing, really, that you can take whatever 15-20 mils of your blood and really we can separate these out into different fractions, and you can do this B for blood here, F is for FICO, which is a reagent we can use, so when we can spin this blood quite quickly, it separates. So this is basically the pre and the post kind of image. Here's what happens when you take blood and spin it, and it separates into essentially three components. Plasma, which is P here, which is essentially the soluble fraction that contains lots of proteins and other molecules that are important in the immune system. And much of what's called your innate immune system is inside here. We'll obviously talk about that. There's this W here, which stands for white blood cells, or white layer, which I presume you can see obviously is white. And that's where really all of your white blood cells are, and really all your immune system, your adaptive or memory-like immune system, resides in that very, very tiny little gap there, which again, is really quite amazing. And obviously R is for red, so red blood cells. So these are obviously the oxygen carriages. So they have no role in your immune system, but obviously quite important to life. So really between the plasma in your blood and the white blood cells, this combination is your immune system. So we'll actually spend some time looking at that. We'll take bits out of these two layers and we'll actually look at what's inside those compartments. But the main part of the labs will be looking at how your body knows that you've been infected, and that may not sound like such a difficult question, but in reality, if I can actually hear my kids out of the garden right now playing. If one of them accidentally, shock horror, cuts themselves on a thorn or something, and they get an infection, maybe there's something on that thorn. How does your body know that that's happened? How does it sense that? Especially if it's not something it's ever seen before, some bacteria it's never seen before. The question is, what do you sense? How do you sense it? And how do you go about trying to resolve it? And that's really what your innate immune system does. That's the one that we all have. It's a genetically encoded one, and it's super important. And so what we'll do is we'll try and understand how, through using biochemistry and biophysics and some maths and statistics, understand how that actually functions. So hopefully, if you see this movie here in front of you, which will maybe be playing, yes. But I hope you can see down here, this one here. This is a macrophage. This is one of the innate immune cells of your immune system. It's finding some bacteria, and that's all these things here. And it's not just finding them; it's finding them and it's eating them, and it's very hungry. It's continuing to gulp all these things here. And what I hope you can see really is that you can see hopefully the outline of the cell. This is the nucleus here. Sorry, about there. Nucleus here. And there's beautiful protrusions of membrane ruffling over this side here. Again, this is classic cell biology, but the question really the labs will address is how does this macrophage know to go in this direction? How does it know those bacteria there? Like, don't forget, you can see them. It can't see them. It doesn't have eyes. Something that's often overlooked through the anthropomorphization of cells is that how does this cell know to move in that direction? So we'll discuss that. What are these bacteria doing to call out this cell? And it does that because the E. coli, which this was in that case there, the bacteria, they send out, without realizing it, a message through a gradient of what's called a chemoattractant. So they have when you're nearer to the bacteria, they produce a large amount of a soluble factor, and this causes a gradient. So the further you are away from it, the weaker that concentration is. And cells like macrophages, like neutrophils, and various other innate immune cells can read that gradient, which means they know to move in this direction. So the question is, we're trying to use both a look at the biochemistry of how you form these gradients, the physics of the formation, and also how the cell biology, how does a cell move in one direction rather than just all over the place, like lots of other cells like to do. So we'll use chemotaxis assays, which will do this by live cell microscopy. So what we'll hopefully see is we shall put the gradient, so a molecule on this side here, and these are the cells in here. And I hope you can all see that in general these cells are moving in this direction, this way, because they recognize this gradient and move along it. So we'll actually try and not only view this on the microscope, but we'll actually try and quantify it as well. But the really interesting thing we're going to hopefully try and do is try and see if we can identify new to science chemoattractants. So what we'll do is we'll go around various labs in Warwick Medical School and ask them for solution supernatants from various bacteria they work on, and just stick them on this side here, and just see if we can make a cell line, which is known to be very good at chemoattracting, move towards this supernatant here. So we're hoping we'll obviously have some controls to make sure it actually works, but we should be able to find hopefully some new molecules or molecules mixes that actually recruit cells of your immune system, which hopefully will be an interesting thing to find out. And obviously, if we do find interesting things, we can move on from that in the future. So that's the plan: is to essentially use a sort of a live cell microscopy screen to find new factors that can recruit immune cells to sites of infection. We'll also try and write some code to see if we can track these cells. We'll try and link into the MATLAB coding you'll do at the beginning of the course and throughout the first year. So hope you can see that these there are some nice trails on these cells here because we're using some mathematical coding to find those cells. And so we'll either do that together, or you can, those who are better at coding already can think about how they might do that themselves. You can obviously then do some quantification of, roughly speaking, the median movement of these cells. So this will again bring in a sort of computational approach to understanding a cell biological problem in your immune system. So just to finish up, I'd just say, try and show you what you can expect to learn during these 2-week block and block 13 on the course. And obviously, you're not going to get to be an expert in the immune system in 2 weeks. It's not something I consider myself to be after 15 years. But the hope really is to give you a real good broad view of what your immune system does and why it's so good at being able to recognize self compared to non-self. So you'll be able to describe the major components of our immune system, both from the cells and the proteins that are also involved. Understand the difference between a genetically encoded or the innate immune system and the learned or adaptive immune response, the one that provides you the memory. So in this case, the innate one, your innate response is one that's really going to recognize whether you have, coronavirus in your throat at the moment, and the adaptive immune response is the one that will hopefully provide you with a memory or an immune response against it for the years to come, so you don't get it again. I'll talk about how genetic recombination is really essential to generating immune diversity. To understand what this kind of use of non-genetically encoded material in your genome allows you to provide almost limitless diversity of recognizing foreign antigens, foreign proteins. We'll look at how HIV and tuberculosis have learned how to evade your immune system, so why they're so good at hanging around when you should have cleared most infections after a couple of weeks. And we'll also discuss what autoimmunity is and what its natural consequences of autoimmunity are. And we'll also touch on cancer therapy as well, because sometimes cancer is also as an issue where your immune system doesn't do its job correctly. So hopefully that gives you an idea of what we're hoping to do over the 2-week block. And thank you for listening. And you can feel free to ask any questions. I'm sure my email address will appear at some point. So thanks very much.